Hello everyone, welcome to our channel, MooTechie. Today's topics: isozymes, regulatory enzymes, zymogens, ribozymes, and enzyme immobilization. Visual heavy, clean, minimal learning experience. Subscribe and share to support free education. Are you ready? Let's begin. First up, isozymes. What and why? Isozymes are distinct forms of the same enzyme. They catalyze the same reaction but have different kinetics and regulation. They show tissue-specific patterns like LDH1 through LDH5. This enables fine metabolic control in the body. The diagram shows the structure from DNA to the functional enzyme. We have genes called LDHA, LDHB, and LDHC. Their mRNA is translated into protein subunits LDHM, LDHH, and LDHC. These subunits assemble into a tetramer, which is the active lactate dehydrogenase enzyme, with different combinations creating the various isozymes like LDH1, LDH2, and so on. Let's look at LDH isoenzymes through a clinical lens. This educational diagram details lactate dehydrogenase, or LDH, isoenzymes, their tissue distribution, and subunit composition. We see the five main types: LDH1, LDH2, LDH3, LDH4, and LDH5. For each, the diagram shows a small icon representing its primary tissue and its subunit makeup. LDH1 is found predominantly in the heart and red blood cells, represented by a red heart icon, and is composed of 4 heart subunits, noted as H4. LDH2 is also in the heart and red blood cells and has a composition of 3 heart and 1 muscle subunit, or H3M1. LDH3 is associated with lung tissue, shown with a blue lung icon, and has 2 heart and 2 muscle subunits, H2M2. LDH4 is found in liver and muscle, with a green icon, and has 1 heart and 3 muscle subunits, HM3. Finally, LDH5 is also in liver and skeletal muscle and is made of 4 muscle subunits, M4. A key explains that the heart subunit is shown in blue and the muscle subunit in gray. So to summarize, LDH1 and LDH2 are primarily in the heart and red blood cells, LDH3 in the lung, and LDH4 and LDH5 in the liver and muscle. The specific pattern of these isozymes in serum aids doctors in diagnosis, for instance in heart attacks, hepatitis, or hemolysis. These enzymes are tetramers formed from combinations of H and M subunits ranging from H4 all the way to M4. Assays that measure these different forms guide critical clinical decisions. Next, we explore regulatory enzymes, the gatekeepers. These enzymes control rate-limiting steps in pathways. They are modulated by allosteric effectors and covalent modification. They often display sigmoidal kinetics, which indicates cooperativity. Their job is to ensure precise flux control and maintain homeostasis. The diagram compares allosteric inhibition and activation. On the left, for allosteric inhibition, enzyme 1 has an active site and a separate allosteric site. An inhibitor binds to the allosteric site, which causes a change in the shape of the active site, preventing the substrate from binding effectively. On the right, for allosteric activation, enzyme 2 also has both sites. An activator binds to the allosteric site, which shifts the active site into a shape that now readily accepts and binds the substrate. This content is copyright MooTechie, educational content. Let's understand allosteric regulation as molecular switches, illustrated by these curves. The diagram again shows 2 enzymes. Enzyme 1 is inhibited. An inhibitor binds to its allosteric site, distorting the active site so the substrate can't fit. Enzyme 2 is activated. An activator binds to the allosteric site, shifting the active site into a perfect shape for the substrate to bind. The key takeaway is that an effector binding to the allosteric site causes a conformational shift in the enzyme. Activators favor the relaxed, high-affinity R state. Inhibitors favor the tense, low-affinity T state.
This creates cooperative S-shaped kinetic curves with a Hill coefficient greater than 1. This mechanism enables rapid on and off control of metabolic pathways. Now, consider feedback inhibition as the built-in brakes of metabolism. Part A shows a simple reaction pathway. A key feature is that the end product of the pathway inhibits the very first committed enzyme. This creates a negative feedback loop that stabilizes the output, preventing too much product from being made. And it improves the efficiency of resource use in the cell. Moving on to zymogens, from safe to active. Zymogens are inactive pre-enzymes that are activated by proteolysis, which is cleavage, by another protease. This prevents premature protease damage to the cells that produce them. Activation is triggered by factors like pH change, specific proteases, or even autocatalysis. They play a key role in processes like digestion and blood coagulation. The diagram shows a classic activation cascade for digestive enzymes. Proenzymes like trypsinogen, chymotrypsinogen, proelastase, kallikreinogen, and procarboxypeptide are all activated. Enteropeptidase converts trypsinogen into active trypsin. Then trypsin itself activates all the others. Chymotrypsinogen becomes chymotrypsin, proelastase becomes elastase, kallikreinogen becomes kallikrein, and procarboxypeptide becomes carboxypeptide. Let's zoom in on two major zymogens, trypsinogen and pepsinogen, and their activation flow. On the left, pepsinogen is activated by the low pH in the stomach plus autocatalysis by already formed pepsin, resulting in the active enzyme pepsin. On the right, trypsinogen secreted into the small intestine is activated by the enzyme enteropeptidase, forming active trypsin. This active trypsin then goes on to activate all other zymogens, creating an enzyme cascade. This cascade results in highly amplified response, where one activation event triggers many more. So enteropeptidase turns trypsinogen into trypsin. Pepsinogen turns into pepsin via acid and autocatalysis.
Now for something fascinating, ribozymes, which are catalytic RNA molecules. These are RNA strands that drive biochemical reactions, like phosphoryl transfer. Major classes include the hammerhead ribozyme, group 1 and group 2 introns, and RNase P. They fold into specific active tertiary structures, often with the help of metal ions. They have exciting uses in therapeutics and are fundamental to our understanding of early life and symbiosis. The slide includes two supporting images. One depicts microbial biosynthesis, highlighting the role of ribozymes and RNA catalysis in cellular processes. Shifting gears, let's talk about enzyme immobilization. First, methods 1. Adsorption and entrapment. Adsorption involves a simple setup at low cost where enzymes are reversibly bound to a support material. The risk here is enzyme leaching, where they can detach. It's suitable for quick applications. Entrapment uses gels, like alginate, to physically trap enzymes inside. A drawback can be diffusion limitations for substrates and products, but it protects the enzyme from the external environment and offers moderate stability. Encapsulation places enzymes inside microcapsules or membranes. This offers excellent enzyme protection, controlled permeability, and enhanced operational stability. When selecting a method, we must evaluate mass transfer needs, consider substrate and product size, balance cost versus performance, and match it to the application requirements. The diagram at the bottom summarizes these methods, under the umbrella of enzyme/cell immobilization methods, listing adsorption, entrapment, encapsulation, and cross-linking. Continuing with immobilization methods 2, covalent binding and cross-linking. Covalent binding creates very stable, irreversible attachments between the enzyme and support. A possible downside is some loss of initial activity due to the chemical process. It's excellent for long-term use. Cross-linking often creates carrier-free immobilized enzymes, like CLEAs, which are cross-linked enzyme aggregates. These offer high reusability and stability without needing a separate support material, making production cost-effective. Ionic binding is a reversible attachment method that is gentle on the enzyme's native structure.
However, it is sensitive to changes in pH and ionic strength. It allows for easy enzyme recovery. Choosing the right method requires matching it to stability requirements, considering throughput needs, balancing cost versus durability, and optimizing for the specific application. The diagram illustrates concepts like adaptation, covalent bonding to carbohydrate or sulfur-based supports, and ionic interactions. Finally, what are the applications of immobilized enzymes? In the food industry, glucose isomerase is used for high fructose corn syrup, or HFCS, production. It allows for cost-effective continuous processing, enhancing product quality and consistency while reducing enzyme consumption. In diagnostics and biosensors, immobilized enzymes provide stable signals for detection. Think of glucose monitoring devices. They offer rapid response times and a long shelf life. In pharma and biocatalysis, they enable stereoselective synthesis steps. Their high reusability over multiple cycles reduces production costs and creates cleaner reaction pathways. In wastewater and textile treatment, immobilized enzymes offer robust operation in harsh conditions. Scalable treatment systems use them for dye degradation and pollutant removal, enabling more eco-friendly processing. Thank you for watching MooTechie. We hope you enjoyed learning about isozymes, regulatory enzymes, and immobilization. Support us to keep education free. Please subscribe, like, and share our videos. Don't miss new videos. Support our channel. Spread knowledge. Thank you. See you in the next class.